So I'd like to welcome Patrick MacDonald from the University of Alberta, and he's going to be talking about a recent review that he and colleagues have just published in Diabetologia on resources for the study of pancreatic islets. So the islets of Langerhans are structures found within the pancreas that are very critical to the pathology of diabetes. I wondered, could you provide initially just a little bit of background on islet research and how it's been used so far to study diabetes?
Yeah, of course. First of all, thank you for asking me to speak to you about this topic. I've been very fortunate to work with a number of wonderful colleagues on this review that we're talking about. And the review focuses on, as you mentioned, pancreatic islets, and these are very important to essentially all forms of diabetes, where the islets are either destroyed through an autoimmune reaction or fail to compensate for insulin resistance. In fact, many genetic contributors to diabetes work by impairing islet growth, structure, or function. And we actually know that the pathology in the islets of Langerhans has been recognized for many years, even before the discovery of insulin, more than 100 years ago. And so we're still learning more about how islets contribute to diabetes and what drives islet cell dysfunction. In the last many decades, the study of isolated pancreatic islets has become possible, and this came about through work in the 60s and 70s on microdissection and isolation of pancreatic islets, through the 80s with the development of automated systems for islet isolations. And so it's been quite a long time now that islet cells have been studied in a dish in isolation. And we have so far gained a great deal of knowledge from this. This includes fundamental understandings of how islets and insulin secreting cells called pancreatic beta cells work to regulate blood sugar. We've come to understand quite a lot about the metabolic pathways that control insulin output and how different hormones or even drugs like GLP-1 drugs, such as the current weight loss therapies, how these regulate hormone secretion from islets, insulin, along with other hormones. Of course, there are many studies as well. Lots of things we've learned about islet dysfunction and islet destruction in type 2 and type 1 diabetes, respectively. And we're starting to learn more and more about the molecular and genetic drivers of these diseases and how some of these genes, these so-called diabetes risk genes, work to regulate islet cell function and survival. I'll even say that it's obviously quite clear that islet transplantation studies have benefited from access to islets for study. In fact, it's progress in islet transplantation through the 1990s, that really set up, I think, where we are today, talking about islet cell resources for the broader diabetes research community.
Right, well, thank you. That's a really good introduction and background. So that brings us on, I suppose, to the kinds of facilities and resources that there are currently for islet research.
Yeah, so when we're talking about human islet biology, the landscape is, I think, somewhat complex and perhaps a little fragmentary at the current time. I think we can break it down into two or three different types of resources for accessing tissue. The first I'll talk about is our biobanking efforts that largely focus on access to high quality fixed pancreas and tissue samples from organ donors with and without diabetes. The clear pioneer in this area is NPOD, the Network for Pancreatic Organ Donors with Diabetes, who have for many years now been leading in this area. But there are also efforts to study live tissue and live islet cells from organ donors. And this can sort of broadly be broken down into two different categories, if you will. There are local initiatives to isolate islets from human organ donors. Often these are tied to the development of pancreas and clinical islet transplant programs. It's a bit challenging to assess how much of this activity is going on and where some of these programs sort of come and go, but nonetheless have been very important for diabetes research. What the review focuses on a little bit more with respect to live tissue are larger initiatives that share tissue, including research islets more broadly. In some cases, these have developed large networks of islet isolation facilities and researchers who are accessing this material. So without going into all of these, they're talked about in this review, but there are notable initiatives in the United States, such as the Integrated Islet Distribution Program. There are programs here in Edmonton. There's actually two here in Edmonton. The Alberta Islet Distribution Program, which is linked to the Clinical Islet Transplant Program, and then a program that I run called the Alberta Diabetes Institute Islet Core, which is focused only on research pancreas and research tissue. And a few notable European programs that should be mentioned as well, led by Uppsala, Pisa, Leiden, and many others. So, I guess what I'd like to say about this is that, access to human samples and islets for research, can be somewhat variable, impacted by things like geography, cost, regulatory restrictions, and so on. I think it's also important to point out that much of what I've just mentioned is centered in North America and Europe. So there, are some important questions that we need to address about disparities in access to human islet research, both within and outside Europe and around the world.
Yeah, so I was wondering because I think your review covers some of the things that are being done to scale up islet research and maybe try to bring it all together. So, are there some initiatives to try to draw this together and look at like data management in order to make things better available to researchers in different areas?
Yes, so I spoke about disparities in access to human
Mm,
islets for research. And one of the ways that this can be addressed and is being addressed is through initiatives that are focused on sharing data.
mm.
So if an investigator, for example, can't have access to live human islets for study, they can have access perhaps to data that's been collected on these samples. Some of the earliest efforts in this area have focused on sharing of genetic and genomic data from human islets. More and more, we're seeing sharing of much more detailed human islet data as well. And this has been wonderful and a great help to the field. In recent years, in particular, there have been efforts in what we would call deep phenotyping of human islets. And these efforts are focused on collecting lots of different kinds of data from human islet isolations. So this could be genetic data, molecular data, such as protein expression or gene expression. This could be functional data like insulin secretion measurements or other hormones that the islets secrete. They can be, data on imaging of these cells and so on. So a very deep and comprehensive phenotyping of islets that some programs are undertaking and making that data available to others. The Integrated Islet Distribution Program that I mentioned earlier has developed a web platform to share islet phenotyping data that they're generating. There's a program in the US called the Human Pancreas Analysis Program, which has been a very important contributor to obtaining and sharing deep phenotyping data from donors with and without diabetes. In Europe, there are efforts like Tiger and Islet GeneView, which largely seek to share genomic characterizations of islet isolations. For my part, I've contributed to a consortium that has resulted in a web platform called humanislets.com, which seeks to share and increase accessibility of data from our own islet isolation program here in Edmonton. I think these resources have been important for a couple of different reasons. Um, they're democratizing access to data, so making data available to be used. Um, but also importantly, they link and combine data sets. And I think that in the future, we're going to see a lot of things coming from that linking of data sets, insights that we may not have been able to make before. But still, some of this remains pretty fragmentary. And there's one last resource that I'll mention, which is still fairly new. It's something called PankBase. And this exists really to start bringing together data from many of these disparate sources and improve usability and improve access.
You talked a bit about disparities in access for researchers, but are there also, would you say, sort of gaps in terms of donor samples, in terms of people of different ages, different ethnicities, social backgrounds, regional areas? Is that also an issue?
Yeah, I alluded to this earlier, and it's something that I think is seen in many different kinds of biobanking programs, data repositories, phenotyping efforts, and that is a lack of diversity in the types of donors or the backgrounds of donors that we're currently able to assess. In part because of the um, geographic distribution of some of these big programs, um, the data currently that is being collected is fairly, centered on European ancestry, for example, North American population demographics, and so on. There really is less or little representation from other uh, ethnic backgrounds, and so there's a problem with the diversity of donors that we're assessing currently. I think beyond that, there are some other issues as well with respect to um, the diversity of donors that we're currently able to collect and study. And this includes different stages of diabetes, different ages of donors, and so on. So if we really want to understand the true, variation in the human population, we need much greater numbers of donors, and we need much more diverse sources of donors to study.
So over the longer term, how do you see these gradual improvements and hopefully this increased networking and sharing of data? How do you see that as being able to improve diabetes research and diabetes care over the longer term?
Yeah, I think there's still some hurdles that we need to overcome. Some things that we haven't really talked about, like standardization between
Mm.
many different islet isolation and phenotyping programs. So the ability to combine data sets currently is fairly limited. And if we are to get to this point of trying to understand some of the bigger questions that require large numbers of people or donors to look at, we're going to have to work on standardization of some of the measures that we're making. The review talks a bit about this, but I think if we can get to a point where multiple centers are on the same page with standardization, data sharing, data integration, then we get closer to having um, a larger number of donors that can the large number of donors needed to address some important questions like, how does the normal variation in genetics and insulin production contribute to the development of diabetes or diabetes risk in a larger population? Um, and this brings me sort of to your actual question, which was sort of what does the future hold in these kinds of programs for their contribution to understanding and treating diabetes? And what I'd really like to see is a move to link what programs like ours are doing, understanding the molecular drivers of cell dysfunction and biomarkers of pancreas biology, to connect those to larger populations and cohorts of patients to understand how this cellular variation contributes to variation in diabetes and treatment outcomes. And we're getting closer and closer to that, I think.
That's fantastic. It sounds like there's a very promising future ahead, and I think that your very, detailed review will garner a lot of interest in diabetologia. So, thank you very much for talking to us.